Question 4. Plants contain many different tissues. Complete the sentences. The leaf tissue that contains the most chloroplasts is the 而家講緊樹葉，樹葉你要知道佢個cross section。咁你上面嗰好多 rectangle 嗰啲 cells，嗰啲叫 palisade cells。The tissue that contains many air space，即係 palisade 下邊好多 gap 嗰個位叫 mesophyll，or spongy mesophyll。跟住 the plant tissue that can differentiate throughout its life is differentiate，即係可以變唔同嘢。咁我哋人入邊就叫 stem cells 啦，咁咪 plant 就叫 meristem。呢啲要記得啦，冇得唔記得。4.2 Xylem tissue transport water through a plant. The walls of xylem contains cellulose. Yes. Name one other substance that strengthens xylem tissue. 咁如果係講緊 support 有兩嘢就係 cellulose 同埋 lignin fibers. 4.3 Phloem tissue transports dissolved sugar around the plant. Name a process that transports dissolved sugars around the plant. 我哋講緊 transport of sugar 唔係 water。如果 water 就 osmosis 啦，咁 transport of sugar 就叫 translocation。唔係 active transport，active transport 係用 energy to move something against the concentration gradient。Figure 3冇 picture 應該就做唔到啦。OK。4.4 我哋做唔到，因為冇咗個 picture。We're going to carry on. Phloem tissue transports sugars to other parts of the plant. The concentration of dissolved sugars in phloem cell in figure 3 is higher than in cell X. Explain how subcellular structures help to move dissolved sugars from cell X into the phloem cell. OK，咁我哋雖然冇咗個 picture 嚟睇，但我畫咗個 small diagram 係講緊呢個 question 嘅。佢就係話 concentration of dissolved sugar in phloem cell in figure 3 is higher than cell X。咁當我呢個係 phloem 啦，咁有個 high sugar concentration 啦，咁呢個 cell X 就係呢度就係有個 low sugar concentration。咁個問題就問 explain how subcellular structures help to move dissolved sugars from cell X into the phloem cell。點樣可以搬啲 sugar from a low concentration to a high concentration？我頭先啱啱講咗係一定要 involve active transport 係咪？咁 active transport 我哋要 energy 啦，咁邊一個 subcellular structure 會 provide energy？咁一定係 mitochondria。OK，咁第一樣嘢要講就係 mitochondria and cell X will do aerobic respiration to release energy。咁呢度至少已經有2分啦。OK，咁個 second part of 個 answer 就係啲 energy 會用嚟做 active transport to move sugars from cell X, which is a low concentration, to the phloem, which is a high concentration。咁你夠寫呢幾樣嘢就攞到5分㗎啦。4.6 The new phloem cells form where unspecialized plant cells differentiate and become specialized. Describe one change in structure that occurs when an unspecialized cell differentiates to form a phloem cell。你記得 phloem 同 xylem 全部都冇 nucleus 㗎嘛？OK，咁你就寫lost of nucleus。Okay, question 5. Student investigated the effect of concentration of salt solution on the mass of uncooked potato pieces。呢一個如果學校有做嗰個 experiment 就係用個 cork borer 嚟 cut 個 potato。OK，跟住你 cut 到個 potato 之後就擺佢唔同嘅 salt solution，跟住你睇下嗰個 potato 有冇turgid 到，就係整大咗啦，或者佢係 flaccid 咗，即係縮細咗。呢個係 change of mass 啦。佢喺普通水佢就0.2 gram，跟住個 salt concentration 越嚟越高嗰陣時，咁佢個 size 佢個 weight 就不停咁 decrease。Plan a method that could be used to obtain results in figure 4。OK，咁我哋 plan 呢個 method to obtain the results。咁第一樣嘢就係要攞個 potato 先啦，咁就 cut the potato into identical pieces，係記得要 weigh the mass of each one。OK，the second mark 就係 prepare different concentration of 嗰個 salt solution。你可以 give example 就0.2至到1，跟住 increase 0.2 every time。跟住 number 3，咁 of course 你要擺嗰啲 potato piece 落去，譬如15 minutes。 跟住你浸完15分鐘，咁就要攞翻出嚟，跟住 make sure 佢係冇晒啲 water 喺個 surface 度，咁一定要 dry using paper towel，跟住就可以 weigh。跟住 part five 你可以 repeat each concentration，至少2次啦，跟住可以 average 你個 results，咁就應該 accurate 啲。Okay, and of course 你每次做 experiment 咁，你就要 keep 有啲 variables 係 under control。咁得6分，我求其寫1兩個應該 OK 嘅。咁有啲咩要 keep the same 就係 size potato must be the same 啦。same time in the solution，我哋如果決定咗15分鐘，就用15分鐘。跟住 obviously the temperature of the salt solution 都要係一樣。如果你寫呢個 method 再加啲 control variable，應該就攞晒6分㗎啦。5.2 explain the results potato pieces in 0.6 molar per dm cube salt solution。如果我哋見到0.6 mol per dm cube 呢個 salt solution，佢係有個 decrease in 個 potato mass by 0.2 grams。OK，所以第一分就係講佢 decrease，第二分點解啲 water 會 leave 個 potato，跟住去個 surrounding sugar solution 就係因為佢個 water potential difference。OK，咁佢入邊個 water potential 就係 high，出邊個 water potential 係 low，咁嗰啲水就 go from high to low。OK，咁睇完個 graph 之後，我就見到個 potato piece 一定 decrease in mass 啦。咁個 explanation 就係個 water potential inside the potato is higher than the salt solution。咁呢個 reason，咁 water will leave the potato via osmosis，即係個 water 嘅 diffusion。5.3 explain why the results potato pieces at 1 molar was different from the result at 0.6。OK for 2分，其實唔使講咁多嘢，你就要 note down 佢係 lost even more mass。個 reason 係因為佢個 concentration gradient 係更加大。Okay, question 6. The question is about pathogens. A scientist investigated antibiotic resistance in bacteria. Name one the antibiotic. 咁佢最 common 嗰個就係 penicillin。The scientist grew one type of bacterium of agar in a petri dish. The scientist placed paper discs, each containing different antibiotic on the agar. The shows the appearance of the petri dish after 2 days. Antibiotic C on paper dish, antibiotic B, antibiotic A, area where bacteria are growing。咁個灰色呢度就有好多 bacteria 啦，咁你好明顯見到佢呢度係冇 bacteria growth。OK，所以 antibiotic B 係 work 嘅。antibiotic C 佢呢度有少少area 係冇 bacterial growth，即係代表佢都 OK，但係唔夠 B 咁勁。A student said the bacterium is resistant to antibiotic C. Explain how the results of the show the student is not correct. OK，一定唔係 correct 啦。OK，呢個係完全唔 correct 嘅，因為你見到antibiotic C 係可以 kill 個 bacterium，因為我哋叫呢個做 zone of inhibition around area C，因為佢根本生存唔到。OK，個 second mark 就係講緊，如果個 bacterium 係 resistant，咁一定會有 growth around antibiotic C，或者你可以講話 there won't be a zone of inhibition。Suggest why doctors are concerned about antibiotic resistance。OK，咁 antibiotic drugs will be ineffective，跟住就殺唔到啲 bacteria 啦。OK，第二分你一定要講係個 diseases will be more difficult to treat，因為啲 drugs 唔 effective 啊嘛。OK，diseases caused by viruses cannot be treated using antibiotics. Suggest why viruses cannot be grown on agar。你記得 how virus work 就佢一定要 invade 咗個 cell，咁佢先可以做佢嘅 reproduction 係咪？咁 A 格嗰度係冇 cells 俾佢 invade。OK，咁呢個係少少 link 住 6.4。OK, 6.5. Why is it difficult for scientists to develop drugs to destroy viruses？你記得個 virus 會喺個 living cell 嗰度，咁你如果要 destroy 個 virus，即係要 destroy 個 living cell，即係你要用個 drugs 嚟 damage 你個 living cell，咁就唔係咁好啦。咁呢個就係個 reason 啦。Question 6.6. Which disease is caused by a virus that damages white blood cell？咁我哋講緊 virus，咁一定係 gonorrhea 同埋 salmonella 呢兩個係 bacteria。 咁 virus 得 measles 同 AIDS 啫喎，咁 measles 唔係 target white blood cells，咁一定係 AIDS，因為佢係 immunodeficiency 就會 attack 我哋個 immune system，即係我哋個 white blood cells。one mark。Student investigated the effects of different factors on photosynthesis. Student used 3 leaves growing on the same plant. Each leaf was treated in a different way. After 48 hours, the student tested that each leaf for starch. Upper and lower surfaces covered with black paper, 咁即係冇 sunlight 啦。跟住 upper and lower surfaces covered and sealed with transparent plastic，咁呢一個我哋冇 CO2 for photosynthesis。OK，如果佢冇 oxygen 就代表冇 respiration。但係今次係 no starch，即係 no photosynthesis，就 CO2 啦。如果呢個唔係 covered，咁即係個 plant 可以 fully photosynthesize，咁一定有 starch present。5分，咁你 explain 翻頭先我講嗰啲嘢就 OK。OK， 7.1，咁我就 put in writing 頭先我講嗰啲啦。Leaf 1就 no sunlight for photosynthesis。Leaf 2 CO 就 no CO2 for photosynthesis。跟住 leaf 3就係 plant can fully photosynthesize。Glucose is made, which is converted to starch for storage。如果你講呢幾樣嘢，咁就有晒5分。記得個 plant 一定會 convert to starch for storage，因為 glucose 係 soluble，就 store 唔到啦。Another investigation, the student used a different type of plant. Plant was left and covered in the light for 48 hours. After 48 hours, the student tested the leaf from the plant for starch. Leaf shows the leaf before it's tested for starch。Green part, white part。OK，因為佢個 part 係 greens，即係代表有 chlorophyll，有 chlorophyll 即係有 photosynthesis。咁如果做個 starch test，咁佢就會係 blue-black。White part 我哋就冇 chlorophyll，咁即係冇 photosynthesis，咁一定會 remain orange。Explain the results you gave in sample。哦，頭先講咗啦。OK，我頭先都講咗 green part of the leaf contains chlorophyll，咁呢個就係 light absorption 啦。咁如果有 light，即係代表有 photosynthesis，咁即係代表有 glucose starch。2分。7.4 Which iron is deficient in a plant with yellow leaves？如果個 plant 唔夠 magnesium，咁佢就會變黃色啦，咁即係 magnesium iron 啦。Give the scientific term that describes yellow color in the leaves。你記住呢個 term 就係 chlorosis。OK，咁即係呢啲 terms 你要背同埋要識串啊，真係幫你唔到。7.6 Rate of photosynthesis is affected by different factors. How could oxygen produced during photosynthesis be used to measure the rate of photosynthesis? OK，因為 photosynthesis 會 produce oxygen，咁如果越多 oxygen produce，咁即係代表佢個 rate of photosynthesis 係越快。咁你可以 count bubbles，或睇個 volume of oxygen release，如果用一個 upside down volumetric tube。OK， light, carbon dioxide and temperature are limiting factors of photosynthesis。Figure 7 shows how the rate of photosynthesis is affected by light, CO2 and temperature。呢度越嚟越多 light，呢度就個 rate of photosynthesis 係 increasing。咁我哋見到 0.4% 個 CO2，咁我哋係會低，但係我哋俾多啲 CO2，佢係個 rate 會高啲。Definition of limiting factor 就係佢會 prevent 個 rate from increasing。Explain the effects of increasing temperature and increasing CO2 concentration on the rate of photosynthesis。OK，for 呢個 question 你要睇翻個 graph，咁要講4樣嘢啦。咁第一樣嘢就係你見到 increase 個 temperature while keeping CO2 the same，increases the rate，即係15同25個 degree，你見到佢個 curve 係高啲，但係個 CO2 level 一樣，所以呢個 increase 咗啦。咁個 explain the effects，咁點解 increase temperature 會 increase rate 咧？就係因為啲 particles，你記得你啲 chemistry 有多啲 kinetic energy to collide。OK， 跟住你就要講 increasing CO2 concentration 會 also increase the rate of photosynthesis，因為而家我多咗好多 reactants，而家我擺好多 CO2 喺度，可以 available for 呢個 reaction to take place。但係最尾個 mark 就係你呢樣嘢加咗，點都會 reach 一個 peak，因為我哋有 limiting factor，所以佢去到一個 point A 嗰度已經全部去晒打橫去。咁我哋有咩 limiting factor 咧？就係個 leaf 入邊嗰啲 chlorophyll 全部已經用晒啦，所以已經快唔到啦個 reaction。OK， 呢4樣嘢就4分。OK， 7.9 Photosynthesis investigations often use a light source。 The spreading out of light from a source obeys the inverse square law. The inverse square law links light intensity to the distance from the light source. Which of the following shows the inverse square law? 其實個 inverse square law 都係講緊 light intensity 係 inversely proportional to the distance，即係代表如果個 distance double 咗乘2，咁個 light intensity 就會 become 1/4。OK 係一個 square 嚟嘅。咁如果個 distance 係乘3，咁個 light intensity 變咗 1/9。咁因為呢個 reason 咧，所以就 1/, 嗰個就係啱啦。